and I was told that the audience would include a vast array of people from math to biology, not to assume much. So I am going to get Maria to help me with that, okay? I'm going to try and introduce things, but go ahead and raise your hand if I'm like saying jargon by accident, and then I can explain my terms, because I want to catch both the mathy people and the biology people in this talk. So there's enormous diversity in how organisms reproduce, and this is a kind of cartoon sketch of the tree of life for eukaryotic organisms. And here we're just talking about different ways in which sexual reproduction is accomplished across the eukaryotic tree of life. But the key point for this talk is that the vast majority of them are sexual, reproduce sexually, at least sometimes. Maybe not every generation, but over time. There are exceptions. The dandelions in my yard are asexual, and that's only found in about 1% of flowering plants. And all of those are of recent origin. There's one clade that has 2 sister species, but other than that, these are all recent origins. Similarly, in animals, less than 0.1% are exclusively asexual, like the famous whiptail lizards, but the vast majority of eukaryotes engage in sex at least sometimes. When I started as an evolutionary biologist, it wasn't clear exactly when on the tree of eukaryotes this feature first evolved. And we were looking for, oh, maybe there are outgroups that haven't yet evolved sexual reproduction. So what's true about them, and how do they differ from the sexuals? So people like John Logsdon and colleagues looked for the key genes involved in meiosis. That's the part of sex where the genome is halved in DNA content. And what they found was that those genes were actually found in all extant eukaryotes, even the ones that are asexual. So rather than having a deep history where there was asexuals out of which emerged sexuals, every single eukaryote evolved from a common ancestor that had already evolved sex. So there is no outgroup comparison. We're all anciently sexual. So then, and that was, we don't exactly know when that common ancestor lived, but somewhere in the 1 to 2 billion year range. And so everything, including Trichomonas and Giardia and others that are predominantly asexual now, have this history of sex. So I'm going to talk a lot about the evolutionary enigma of sex, and that is this puzzle that sexual reproduction evolved early, really early, is rarely lost entirely, and has been maintained across most eukaryotes. But it's an extremely expensive process, as I want to next argue. What are the expenses of it? So it's this costly process that's been maintained. So let me tell you about the costs. Some of them are kind of obvious, and some are more subtle. The costs of producing males or dividing up resources between sons and daughters, or between eggs and sperm. And just as a cartoon example of that, imagine you have females and they're either asexual and can invest all of their resources into daughters, or they're sexual and they divide up their resources 50-50 between sons and daughters. And in this cartoon where every female makes 2 offspring, so the females reproduce, she has 2 daughters, so the sexual female reproduces, she has 2 kids, but one of them is a daughter and one of them is a son. And then this repeats for those daughters, and they produce granddaughters. Again, every female asexual investing fully in daughters, and sexuals divvying up resources. But you can see where this is going. Basically, asexuals have double the growth rate of sexuals. So sexual females, by dividing resources between daughters and sons, or between eggs and sperm, should be quickly outcompeted by asexual variants, all else being equal. And this isn't a minor selective difference, this is a huge selective difference, like 2-fold in this extreme case. By all else being equal, I mean things like there can be ways around this dilemma, like for example, if fathers are providing paternal care to the offspring or resources to the female, in which case there can be that can equalize the cost. But for a lot of organisms, the cost of sex is thought to be substantial. And that's just cost number one. Another cost that we're going to talk more about today is a more subtle one, and that is that sex breaks apart the very gene combinations that allowed parents to survive, to reproduce. So an organism that is about to reproduce knows only one thing, and that is that its genome works in the current environment in which it's lived. There's information, content in the fact that it's survived to reproduce. By analogy, I'm going to make a poker analogy. So if you are a surviving hand in a tournament and you have either 3 aces or a straight, that's because those cards worked well under the rules of poker. You survived. Just like a genome, the genes work well in that particular genome and those organisms reproduce. So if you were a winning hand in poker, would you want to trade cards with another person? You'd be like, no, I'm holding on to my hand because this 5 of diamonds is really, really not a great card on its own. It's just great in that particular gene combination. So the long and short of that is that individuals who have survived selection are likely to have genomes that work well in the current environment. So asexual reproduction preserves that combination. Sex mixes it up. And that's a cost. We often talk about this as a load, the load of recombination or the load of segregation mixing up those cards. And in addition, there's the cost of finding a mate, the risk of not finding a mate, the risk of disease transmission during mating, the risk of predation during mating, and it goes on. So what pays all of these costs of sex? Now, in your mind, if you've heard of this topic at all before, you're probably like, well, it's obvious, you need variation. And so in some fashion or other, most explanations of sex rely on the idea that sex can generate variation needed by selection through recombination and segregation. So my first task is actually to break this down and say it's not that easy. So that's what I'm going to start. But before I do, and here's an example quote of this our idea. This is from Weissman. So in the 1800s, before we even knew about the genetic basis of heredity, and he wrote, the communication of fresh ids to the germplasm implies an augmentation of the variational tendencies and thus an increase of the power of adaptation. So he didn't know how inheritance worked, but he knew and argued that that combination from mom and dad was important in variation. And that variation was the be all and end all of evolution and adaptation. Okay, so now I want to break this down. And I'm going to do it with kind of simple case examples, but it's a broader issue. First of all, sex doesn't necessarily increase variation, and I'm going to explain why. And then second, not all variation is helpful for selection anyway. So there's kind of 2 strikes against this explanation, and then that makes it a little bit more nuanced. So here are my cartoons. I'm going to go through a few cartoons like this, but this is just illustrating the point, and it's a broader concept. So sex need not increase variation. That happens because selection itself can generate and maintain variation. And when that's true, sex can actually reduce variation. So in this simple case, I'm going to have just one gene underlying some trait, height. And so there's just different genotypes, and the fitnesses are such that the extremes are pretty fit. And so what happened, like maybe these ones can run away from a predator, maybe these ones can hide in a little hole, and these ones are the ones that get eaten by the predators. So after predation, then we have a lot of variation in height. We've got the short ones and the tall ones, and not many of the intermediate ones. So what's going to happen when this population reproduces? If it reproduced asexually, then that variation in height would be maintained. If it reproduced sexually, randomly mating, then it would return to Hardy-Weinberg proportions. Everything would get mixed together, and you would have what you'd expect by binomial sampling of genes from the parents. And the variation is lower. This is less variable than with asexual. So sex does not increase variation. It depends on what selection is doing. Now I'm going to go to the second problem. Not all variation promotes evolution or promotes selection. I'm going to flip the fitness around and otherwise imagine the same scenario. But now the heterozygotes are doing really well. So after selection happens, those heterozygotes, because they have high fitness, they're the survivors. And so you have a lot of them. What's going to happen when this population reproduces? Well, again, if it's asexual, we're going to maintain basically a carbon copy of the parental population. And if it's sexual, then these alleles are mixed up. Again, like binomial sampling, we call it Harding-Weinberg in biology, and you get this population. But remember, so yes, in this case, selection depleted variation. Sex can regenerate it. But who's it making? It's making dead kids. So in this case, variation is augmented by sexual reproduction, but the types of offspring that are produced are the ones that have low fitness. That is, sex is undoing exactly what selection just did. Selection boosted fit genotypes, and sex tends to destroy them, just like the poker problem. After survival, the hands, the genotypes that work well together are surviving. Mix them together and all bets are off. So again, I mentioned that it's called a segregation or recombination load in the literature. Okay, so that clarifies. It's more nuanced. It's a harder problem because to solve this evolutionary enigma of sex, we need to know under what conditions sex actually generates useful variation, missing variation, and that the variation it produces don't immediately just die or have low fitness. And furthermore, we have to ask whether or not those conditions, so these are the conditions under which sex is useful and then helps adaptation. Moreover, we have to ask, are those sufficient to increase the amount of investment in sex over evolutionary time? So those are two slightly different problems. One is asking here and now, what's the outcome of sexual reproduction? The other is, will evolution favor more sex? Just like for those of you who are not in biology, I've got a little bit of a reminder of terms that I need to use in the subsequent slides. So the first thing I need to teach you about is linkage disequilibrium. So linkage disequilibrium measures associations in the population currently. What genes or alleles, variants are found together on chromosomes. And those are associations. So this is my fictitious chromosome. This is an allele that's good on this particular site. This is an allele that's good on that particular site. And so we want to know, when do you get two happy faces together, for example? So if the population is composed primarily of a lot of these chromosomes and a lot of those chromosomes, then there's a correlation or covariance. And you get more often smiley, smiley, and neither, neither. And that we say is a positive disequilibrium. We measure disequilibrium with this particular measure, and that can be shown to be equivalent to a covariance. So are you more likely to carry the smiley, smiley together than you'd expect by chance based on the smiley frequencies independently? So disequilibrium measures covariance. And of course, you could have the opposite scenario where these chromosomes are more abundant in the population than expected. And then you have negative disequilibrium. There's good alleles tend to be found on a bad genetic background. Okay, so in this case, variation is missing because we don't have the really high fitness chromosomes, nor do we have the really low fitness chromosomes, variations depleted. So that's the kind of scenario that we would predict would favor recombination by mixing these overabundant types and regenerating the rare types that have a lot of variation. The second concept that I need to introduce is recombination. So this is a cartoon of meiosis, where you have the chromosomes inherited by this individual's mother and father that have duplicated prior to meiosis. Then they undergo meiosis and make the haploid, so a single sperm or egg type. And then we pay attention to whether or not what's passed on to the offspring are the same chromosome that this individual got from its parent. Then we call it parental chromosomes. These two are the parental chromosomes. Or they could have undergone recombination between those two genes and make the non-parental or the recombinant chromosome. So this is an aspect of meiosis. This little r represents the total probability of recombining and making non-parental offspring type. And you can think of it as shuffling the deck, because it's shuffling the types of combinations that are found in a population, shuffling those genes. Any okay at that point? Okay. So just to emphasize, D is a covariance or association measure. R is a measure of physical distance that's equal to the number of recombination events expected between two sites. Okay, now you're ready for some modeling. So the first point that I want to make is that if you track populations and you allow them to go through meiosis, but also experience selection. And here I'm using the simplest case where it's selection in a haploid. So you can think of this as the big A and big B as that smiley face, and these are the unhappy faces together. And so they all have different fitnesses. The W's are different chances of surviving. And when you crunch through the model, you find that recombination only enters if there is disequilibrium present, which makes sense. If it's already randomized and D is 0, there's no covariance structure, then it doesn't matter if you mix it up anymore. So these are always intertwined concepts, but recombination is breaking down the disequilibrium, the associations that have been built up by past selection. Okay, so that's kind of a starting place. But now the second thing I want to introduce to you is another concept. So this, we can crunch these evolutionary models, but how do we even ask whether sex evolves or more recombination evolves? That can't be done in this model. So the types of models that we construct to examine questions like the evolution of features of organisms is to imagine other genes called modifier genes that alter processes of interest. So for example, we met a modifier gene in yeast that makes it more likely for a cell division to be meiotic rather than budding, asexual. Inducer of meiosis is such a gene. Those genes can influence the tendency to be sexual or asexual. So a modifier, we put that into our model and ask if a variant arises that's more prone to sex, when does that spread or less prone to sex? Similarly, when we're modeling changes in the recombination or the map distance, we imagine a gene, perhaps one that's inducing double-strand breaks or cutting up the DNA in different ways. And we ask whether such genes will spread and increase the rate of recombination or other variants that decrease the chance of recombination spread. So that's kind of lesson number one. We understand not just how evolution acts when there's selection going on, but we can also answer these kind of meta questions about how does the organism and the system, the biological system itself evolve to change that stage on which selection is acting through these modifier models. So just as an example, I'm going to go through a couple of types of models. For example, imagine we had only that one selected locus, the big A and little a, but we altered the amount of sex through a modifier at a second locus, the big M, little m. So we just do a lot of bookkeeping. It's like a lot of just tracking things over time. We track every chromosome as it passes through the life cycle, accounting for how the modifier changes its tendency to be asexual versus sexual. So we write all the equations, we account for mutation, we account for selection, and we account for random mating. I have suppressed most of the equations here. Here's another model. I haven't suppressed the equation. So this is a simple possible model of recombination where you have only going to have selection on the haploid stage. That means everybody has only one copy of every chromosome. But we have to have 3 loci. We have to have A and B between which recombination occurs, and we have to have one of these modifier genes that alter that recombination rate. So I want you to memorize this. No, but I do want to point out it's just a complicated set of nonlinear equations, and but they're just equations. And what we do in this literature is use math to figure out under what conditions will a new modifier gene invade? What's its growth rate when rare? We find equilibria or dynamical systems, and we ask under what conditions will something in boosting the amount of sex spread to a higher level. So this one slide is going to do a lot of work for me, because all I want to say is we did a lot of models. We did a lot of models over decades, and they didn't work well. So what did we look at? We looked at a whole bunch of kind of models where selection, rather than generating variation, tended to deplete variation. So these have different names like synergistic epistasis or negative epistasis or negative dominance interaction. Basically, different scenarios under which selection itself depletes variation. But when we looked at whether or not that favored more sex or more recombination, it was like, only if you balance everything just so. And the reason was that the poker problem is that if selection is depleting variation, yes, more sex and more recombination regenerates that variation, but those are typically dead ends. They're lower fitness types. So only under narrow conditions does this work. You have to have negative epistasis. It has to be weak, not strong, and you can't have variation between your genes and how they're interacting. So not a very compelling explanation for sex across the tree of life. Another commonly touted argument is, well, maybe it's not that selection depletes variation, but maybe selection changes. And what lets your kids survive might not have been what allowed your ancestors to survive. So the world is changing. That's sometimes called the Red Queen hypothesis from the Lewis and Carroll scene, where they're running as fast as they can just to stay in place. And the Red Queen says, well, that's just how it works around here. This is a metaphor for parasites or diseases, or competitors are constantly evolving, and evolution of any host species is just keeping pace. But when we modeled these sorts of host parasite or competitor models, they also didn't generally favor more recombination, more sex, for the very same reason that what helped your parents survive in the current environment of parasites and environments tended to help the kids. And so it was more harmful to recombine. And only if the environment changes often and the associations you need flip, like every 2 to 5 generations does this work. So it's really, yes, maybe occasionally, but it's not a really compelling argument for all eukaryotes undergoing sex So what the heck was going wrong with all of these models? They are the biological equivalent of a spherical cow, is my argument. They were deterministic, and they actually missed the boat. So we have a long history of stochastic models in evolutionary biology, but those stochastic models, the random genetic drift, we think of them as kind of inducing a little bit of noise, just making trajectories of selection wobble. But we don't tend to think of them as changing the answer. And this is a case where including stochasticity changes the answer. So Felsenstein in 1974 pointed out this, he read the literature and actually pointed out there seems to be a pattern when we compare those papers which found an advantage to recombination with those which did not. And he pointed out that depended on this. You assume the population is infinite in size and deterministic, in which case you don't tend to see an advantage to recombination, or it's finite, in which case you do. So perhaps the advantage of sex comes from the challenges faced in finite populations. And I think one way to think about this is just to focus on the combinations that are possible, given you've got 4 base pair alternatives and millions or billions of base pairs. There's a lot of genetic variants that are possible that are going to be absent in any finite population, no matter how large. There's just so many combinations that are possible. So any finite population has a limited number of the combinations, and moreover, selection rapidly depletes those variants. So I'm going to go up and then come back. So it's a combinatoric problem. And when I say selection rapidly depletes the variability that's there, imagine these are just chromosomes with some good and some bad alleles. Selection acts, selection acts, selection acts. And over time, the best of the initial population has risen to high frequency. Those are the ones with 2 pluses. There's very little difference in fitness anymore. They're all about the same, 2 pluses, 2 minuses, but there is still genetic variation. There are different individuals carry the pluses and minuses at different loci, but that's stuck if there's asexual reproduction. This genetic variation is hidden unless recombination and sex shuffle things. But the key is that in a finite population with selection acting, there's going to be kind of a concentration of mediocre chromosomes that can still potentially reveal genetic variation if sex and recombination happen. So this is just another way of saying it with happy faces and smiley faces and sad faces, and then a whole genome can be thought of as a tied together assortment of good and bad combinations of genes. And to the extent that populations are asexual, that comes as one unit. That is selected as one whole mess. And you can't think about selection acting on one site in isolation of the other. There's enough sex and recombination. So you might think, so this idea is, I'm going to call it the selective interference hypothesis for sex, that selection at one site interferes with what goes on at neighboring sites in the genome. And it's actually, it has threads going about a century ago. So Fisher wrote in 1930, for unless advantageous mutations occur so seldom that each has had time to become predominant before the next appears, they can only come simultaneously in the same gamete by means of recombination. So you carry a beneficial allele and you carry a beneficial allele, but they're in different individuals. They'll never come together unless there's sex. Now, Fisher didn't mention drift or finite population sizes, but of course, if the population were infinitely large, somebody would already have that combination. Those mutations would have occurred in the same individual. Muller phrased it similarly: without sexual reproduction, the various favorable mutations that occur must simply compete with each other. But again, that assumes a limited set of possible combinations. I think I'm going to skip these graphs and just go on. Yeah, I think I'll even skip this one because I want to just hammer this home with one example of the problem of selective interference. And that's from a paper looking at recombination and the deleterious hitchhiker. Next one. So an important benefit of recombination is it's untying those bad combinations that appear by chance. So this paper was looking at the following. You have a new beneficial mutation, it happens here. And it's on a chromosome, and somewhere nearby is going to, if you go far enough, it'll have a deleterious mutation segregating in another gene. And because it's finite, this beneficial mutation happens once in one particular background. It's not happening across all backgrounds. And so the question we asked is, when will its fixation, the fixation of the beneficial allele, cause the fixation of this neighboring deleterious allele? And so, so we modeled this. Those are the wild type alleles at both loci. Those ones are just normal. This one's better. This one. So, and recombination can untie them. I'm going to tell you, so why I wanted to introduce this is just to show you the types of mathematical models that we use in this area, and when the stochastic models that we use. So we want to know, when will recombination uncouple the alleles? Just as a little bit more notation, we need fitnesses. So this is the fitness of each of the individuals carrying these different chromosomes. This is the wild type. This one has just the deleterious allele, so D for deleterious. This one only has the advantageous allele, A for advantageous. And then we assume here additivity if you have both. Okay. And the initial state of the system is we just have this one beneficial allele occurred, so 1 over the population size is its starting frequency. And pretty much everybody else is the wild type, Yes, there's deleterious mutations, but they're rare in the population. So for now, we'll ignore those. And we'll ignore the one that has not happened yet, which is the good allele at both populations. So we're going to just simplify it down to this starting condition. And then if there's never recombination, we actually know exactly how this one will rise in frequency. As long as this benefit is greater than this harm, it'll rise in frequency, and we know how. But what we want to know is, what is the chance of a successful recombination before that fixation happens? What is the chance that a recombination rescues the wild type allele from, recombines and allows the wild type allele to recombine into that chromosome with a beneficial allele? But this is just tracking. We have a chance that recombination occurs. We have when recombination happens, it could either create the good, or it can create the bad. We only care about the good. And then we use branching processes to ask, when it occurs, what's the chance that that recombination actually isn't lost in the first few generations after it appears? And that branching process result is this one. It depends on the strength of selection. Anyway, we just model all those, put them together. And then that gives us, if that beneficial mutation appeared, what's the chance that at some time 0, what's the chance that there's a recombination event that establishes at time t? And then we ask, what's the chance that that happens sometime before the fixation, before p hits 1? So this is just taking that and saying it has to happen before, in some individual in the population, and then we integrate that over all generations, and anyway, we get a solution. And it's approximate, when selection is weak, you can approximate and then get an approximate solution for the hitchhiking event. So that's one way. It's we do this all the time in evolutionary biology. We kind of approximate processes, and we use a combination of branching processes where needed, and these deterministic approximations. But there's another way to do that here, which uses diffusion models. So diffusion models are more explicitly finite population dynamics. And here we're interested in the diffusion process between when the wild type alleles were present and this new beneficial allele with this deleterious background. What we want to know is what is the chance that as it rises to fixation, that that it's not killed by a recombination event. So diffusion with killing is a particular type of diffusion process where the killing refers to a stopping condition. So we basically say, this is a diffusion process. That's easy. That's a normal diffusion on one variable, repeat. But to stop this process, if ever a successful recombinant appears. And so if that happens, it's killed. And so there's good analytical frameworks for figuring out that killing probability. And you get formula like this, which with weak selection can be approximated. It's not that different from what I showed before from the kind of first principles explanation I described before. This is the only difference. And N is the population size, so that's likely small. So here are numerical and analytical predictions for population size. This is the advantage of the allele multiplied by the population size. That's the disadvantage of the allele SD multiplied by the population size. And you can see that the probability that this deleterious allele fixes in the population, despite the fact that it's bad, rises the faster that sweep happens. That's this. And in addition, if you change the amount of recombination, it does what you'd think. If there's less recombination between those two sites, it's much more likely to fix before a recombination event. It saves the wild type allele. But the more recombination, the more likely it is that that allele will spread to fixation untied from that close association with a deleterious mutation. So for this, just one question, the probability of a hitch declines exponentially with a recombination rate. The faster that beneficial allele sweeps, the higher SA, the more likely a hitch, the worse the load, the less likely a hitch. And we gave a rule of thumb for when there would be about a 50-50 chance that that hitchhiker would get in. But I haven't mentioned the evolution of sex, the evolution of a modifier. We did another project looking at, well, will this in fact favor modifiers of sex? And in this particular case, the fixation probability of a modifier that increased sex or recombination is shown here. So it goes up and up and up. There's only deleterious mutations, but here we have the problem that we were just talking about, beneficial mutations appearing and getting tied together with deleterious mutations. So with every one of these events that's happening, there's a bit of a boost to sex in the population. So sex and recombination can reduce that selective interference. But then this kind of raises a conundrum. That was just a simulation result. And so now we have a conundrum. Those equations I showed you, I wanted to show you them because they're nasty without a stochastic element to them. You know, like 8 coupled nonlinear equations, and you're kind of lucky to get anything out of it. And so, but now I'm saying, well, but we need to do more stochastic models in order to get the evolution of sex. And we have over the decades made analytical progress with this type of question. And so the types of models that we've used, Nick Barton in 1995 used a multi-type branching process just to show how the probability of fixation of one beneficial allele depends on its background. I added a modifier to that model. So it's again a multi-type branching process model, but your chance of switching types in the multi-type branching process depends on your current modifier genotype. And And that was able to show that modifiers that increase recombination get a boost, are more likely to rise, more likely to fix, because the chromosomes that they generate are more likely to fix, are more likely to untie that deleterious hitchhiker from the beneficial. Another approach that we've used is following the moments, following the mean and the variance, and track covariance between all of these chromosomes. And showing there too that there's a benefit to sex that's emerging only from the finiteness of the population. And then another paper Rose and Michaud did a similar thing with a different, we focused on recombination, and this one focused on segregation at the locus. So just as an example, this is simulations of the multi-type branching process model. No, this is the moment-based approach. I think a decent approximation. So this is saying through this process, one bout of selection of this one set of sites, more modifiers that increase the amount of sex or recombination are spreading. Where we can get analytical headway, though, it's a little bit of a mismatch. We have to assume weak selection, pretty large population size, pretty frequent recombination. So there's a little bit of a, yes, we can get some analytical headway on this, but it kind of tends to reduce the size of the effect. But the key point is, this is what we'd see, the solid line is what we see in an infinite population under the conditions of these situations. There'd be no advantage to sex if the populations were infinite. Yeah, 2 more minutes, okay. Selective interference is important in larger populations. As you add more loci, add spatial structure, we can talk about this. The other thing I really want to emphasize is, I mentioned that those deterministic models were nitpicky. You had to have everything just so. So in an infinite population, what I mean is you have to have those fitness surfaces be curved in just the right way. And I cherry picked these ways to have the right positive conditions. But what you can see in finite populations is that it doesn't matter what exactly the nature of those fitness curvatures are. So it's much broader and less sensitive to that assumption that epistasis is weak and negative. Okay, I think I'm going to skip over. This is a slide to just point out. Notice that this view of the smiling faces and barky faces across the genome doesn't actually depend on the nature of selection acting. It could be host-parasite interactions at one site, could be deleterious mutations in another site, could be beneficial mutations or local adaptation. It's the net effect of a whole genome that's subject to different forms of selection that is, I think, the powerful force selecting for sex and recombination. And I think with that, I will stop there. Or we can talk about this slide if we want. They're just the best evidence, I would say, for selective interference. But let's stop there and take questions.
Questions? Emphasize the importance of finite versus infinite populations, but also made it clear that there is, with so many base pairs and so that it would seem to me that the infinite
It's pretty infinite.
At the extreme.
Yeah.
So in order to, because the argument is that in an infinite population, you can get the right combinations without a recombination. But is that realistic? You know what I mean?
No.
Right.
So no population is infinite enough for this problem to go away.
Right. So then why draw attention to that distinction between infinite and?
Because modelers, it's so much easier to do the infinite population case. That's our spherical cow. It's a lot harder to do these kind of branching process or method of moments. You basically take your number of equations and you square it, right? Like 64 equation kind of thing to get all the covariances, the variances. So So it's nasty, but avoiding it was not getting us the answer. And so I think the more general point is, where is that going to be true? Where are we going to get the wrong answer by doing deterministic models in biology? And this is a really nice example of that problem. But you know, John, when I started on this, I was like, oh, this is going to be important in populations of size 1,000, 10,000. But this can't explain things in Drosophila or, you know, large. And so realizing that, no, actually, it gets worse in larger populations because they maintain more variation. So there's more of those sites that are variable, polymorphic in larger populations. Problem doesn't go away.
You only briefly mentioned it in passing, but I was wondering about I guess the issue of the number on loci, or I guess figuratively genome size. And I guess my intuition is that the number of hitchhikers probabilistically is going to go up faster than the number of beneficial alleles. Depending what you think of distribution.
Yeah, great questions. And so we try and measure the input rate of deleterious mutations. It's a capital U parameter, and that there's experiments that are trying to estimate that. And though that does scale some with genome size, but it also scales with other things like the generation time and just how error prone replication is. But yeah, the higher that raining down of deleterious mutations, the more important this will be.
30 questions online.
Any questions online? There's no chat, but
Yes, I would like to make a comment and not ask a question. Actually,
you guys can see that.
I want to congratulate you, Sally, on a wonderful talk and express my happiness at how well you convey the importance of stochasticity in your result. So thank you very much.
Oh, thank you, Cindy. That's wonderful to hear you say. I really appreciate that.
Okay, on that very positive note, let's next speak one more.